Polysaccharides. Polysaccharides, also known as glycans, consist of monosaccharides joined together by glycosidic linkages. Polysaccharides that are polymers of a single monosaccharide are called Homo Polysaccharides. Those made up of more than one type of monosaccharide are called heteropolysaccharides. So one is the Homo Polysaccharide, another one is the heteropolysaccharide. Homo Polysaccharides are also classified on the basis of their monosaccharide units. A Homo Polysaccharide consisting of glucose monomeric units is called a glucan. One consisting of galactose units is a galactan. Three important polysaccharides of glucans are starch, glycogen, and cellulose. Starch is the principal food reserve of plants. Glycogen functions as a carbohydrate reserve for animals, and cellulose serves as structural material in plants. Now start with starch. Starch occurs as microscopic granules in the roots, tubers, and seeds of plants. Corn, potatoes, wheat, and rice are important commercial sources of starch. Heating starch with water causes the granules to swell and produce a colloidal suspension from which two major components can be isolated. One fraction is called amylose and the other amylopectin. Most starches yield 10 to 20% amylose and 80 to 90% amylopectin. Amylose typically consists of more than 1000 D-glucopyranoside units connected in a linkages between C1 of one unit and C4 of the next. So this is the C1 and this is the C4. So this is the 1,4 connection. So alpha 1,4 glucosidic linkage. Chains of D-glucose units with alpha glycosidic linkages in amylose tend to adopt a helical arrangement. So this becomes a large chain, this one, this one, this one, this one, this one. So this actually adopts a helical arrangement, and this is the helical arrangement of the glucose units in starch. Amylopectin has a structure similar to that of amylose, that is alpha 1,4 linkage, except that in amylopectin the chains are branched. Branching takes place between C6 of one glucose unit and C1 of another and occurs at intervals of 20 to 25 glucose units. So after 20 or 25 glucose units, you can find a branching in case of amylopectin, and the branching is occurring between the C6 and C1 of these two glucose unit. Physical measurements indicate that amylopectin has a molecular weight of 1 to 6 million. Thus, amylopectin consists of hundreds of interconnecting chains of 20 to 25 glucose units each. Glycogen. Glycogen has a structure very much like that of amylopectin. However, in glycogen, the chains are much more highly branched. Methylation and hydrolysis of glycogen indicate that there is one end group for every 10 to 12 glucose units. Branches may occur as often as every 6 units. Glycogen has a very high molecular weight, almost as high as 100 million. So this is a very high molecular weight, 100 million. Okay, million means 10 to the power 6 and billion means 10 to the power 9. Don't forget these two terms, million and billion. The size and structure of glycogen beautifully suit its function as a reserve carbohydrate for animals. Its size makes it too large to diffuse across the cell membranes. Thus, glycogen remains inside the cell, where it is needed as an energy source. It is a ready source of glucose when cellular glucose concentrations are low, and able to store glucose rapidly when cellular concentration of glucose are high. So this is the structure of glycogen, and this is the branching. It occurs after the interval of each 5 to 6 units of glucose. So this is highly branched unit, okay, highly branched molecule. Animals store energy as fats, which is triacylglycerols. So fat is actually triacylglycerol, and as well as glycogen. So animals store energy as fat and also glycogen. Fats, because they are more highly reduced and capable of giving much more energy. The metabolism of a typical fatty acid, for example, liberates more than twice as much as energy per carbon as glucose or glycogen. So the energy is actually twice than glucose or glycogen in case of fats. But glucose is readily available and is highly water soluble. As a result, it diffuses rapidly through the aqueous medium of the cell and serves as an ideal source of ready energy. So because of this, glycogen is also needed. So animals have two energy sources. One is fat, which is actually triacylglycerol, and another one is glycogen. Long chain fatty acids, by contrast, are almost insoluble in water, and their concentration inside the cell could never be very high. On the other hand, fatty acids, because of their caloric richness, are an excellent energy repository for long-term energy storage. That's why animals have two types of energy sources. Now a question may arise that why can't amylase digest glycogen? Amylase is an enzyme that breaks down starch in the form of amylopectin and amylose. Both amylose and amylopectin are formed by alpha glucose joined together. That means the glycosidic linkage is alpha by 1 to 4 and 1 6 1 4 and 1 6 glycosidic bonds. Glycogen being a highly branched molecule, it is evident that amylase won't be able or won't be good enzyme to digest it due to physical structure constraints. So because of this, because of the physical structure, glycogen is very much highly branched. That's why the amylase can't just digest the glycogen. It is hydrolyzed by glycogen polyphosphorylase. Glycogen phosphorylase. Actually, this glycogen is hydrolyzed by glycogen phosphorylase. I can't utter this. Glycogen phosphorylase rather than amylase. Cellulose. Cellulose contains D-glucopyranoside units linked in 1,4 fashion in very long unbranched chains. Unlike starch and glycogen, however, the linkages in cellulose are beta-glycosidic linkages. So in cellulose, the linkages are beta linkage. The beta-glycosidic linkages of cellulose make cellulose chains essentially linear. They do not tend to coil into helical structures as do glucose polymers when linked in an alpha 1,4 manner. The linear arrangement of beta-linked glucose units in cellulose presents a uniform distribution of hydroxyl groups on the outside of each chain. When two or more cellulose chains make contact, the hydroxyl groups are ideally situated to zip the chains together by forming hydrogen bonds. Zipping many cellulose chains together in this way gives a highly insoluble, rigid and fibrous polymer that is ideal as cell wall material for plants. So this is the two cellulose unit which are coming in contact with each other, and these hydroxyl groups are being zipped with the because of the hydrogen bonding property. There is another interesting and important fact about cellulose. The digestive enzymes of humans cannot attack its beta 1,4 linkages. Hence, cellulose cannot serve as a food source for humans, as can starch. Cows and termites, however, can use cellulose as a food source because symbiotic bacteria in their digestive systems that produce beta-glucosidase enzymes. Thus, the special stereochemistry of glucose allows it to form helical structure when it is alpha-linked, which is in case of starch, and this gives a rigid linear structure when it is beta-linked in case of cellulose. Now come to cellulose derivative. A number of derivatives of cellulose are used commercially. Most of these are compounds in which 2 or 3 of the free hydroxyl groups of each glucose unit have been converted to an ester or an ether. This conversion substantially alters the physical properties of the material, making it more soluble in organic solvents and allowing it to be made into fibers and films. Treating cellulose with acetic anhydride produces the triacetate known as arnyl or acetate. It is used widely in the textile industry. Cellulose trinitrate, also called gun cotton or nitrocellulose, is used in explosives. Rayon is made by treating cellulose with carbon disulfide in a basic solution. This reaction converts cellulose to a soluble xanthate. So if you take cellulose and just mix with carbon disulfide in basic medium, this cellulose will form cellulose xanthate. The solution of cellulose xanthate is then passed through a small orifice or slit into an acidic solution. This operation regenerates the OH groups of cellulose, causing it to precipitate as fiber or a sheet. So at first you are taking cellulose, then forming a cellulose xanthate, then again you are hydrolyzing it to get back the cellulose unit. This is actually the rayon or cellophane. If you get the fibers, those are called rayon, and if you make sheets from it, then those are called cellophane. So the sheets after softening with glycerol are called cellophane. A process called sizzing chemically modifies the cellulose in paper. As a result, the paper resists wetting. In addition, sizzing leaves the paper in a slightly alkaline state. Acid-free paper lasts much longer than paper that is not acid-free. One sizzing process involves treatment of cellulose with 2-alkyl succinic anhydrides, where the R and R' are short alkyl groups, for example ethyl or propyl groups. These are the ethyl or propyl groups,